right, good afternoon, everyone. I am Nawshin Alim. I'm from the University of British Columbia, and I work under the supervision of Dr. Thomas Beatty. Some of you might have heard about him. Welcome to the final talk of the conference, which involves the receptors of the phage-like particle called gene transfer agents in the bacteria Rhodobacter capsulatus. So maybe an overview of what gene transfer agents are. So GTA particles are officially classified as mobile genetic elements, just like transposons, piosins, and plasmids are. They're believed to have descended from tailed phages or codovertices. And we believe that they evolved in prokaryotes to serve the function of horizontal gene transfer in said prokaryotes. This is a nice diagram in a Nature Reviews paper that illustrates the main differences between GTAs and phages. So GTA genes are intrinsically, sort of permanently part of the genome of the producing cell, just like any of its other genes. Whereas we know that phage genes can exist as prophage within the genome of the host cell, but could also exist independently within the host cell. The expression of GTA genes is entirely regulated by the producing cell, by its own genes, whereas the expression of phages is usually regulated by phage genes themselves. And lastly, GTA particles show no bias towards packaging their own DNA within their heads, whereas phages, as we know, predominantly package their own DNA into their heads. So the receptor of the Rhodobacter capsulatus gene transfer agent is still unknown, and that's what my project is about. So diving right into the structure of the RCGTA particle, it has a classic tailed phage morphology, but I'll focus on the receptor binding proteins of the RCGTA particle. So it has, on its head, it has head spike fiber proteins, as many phages do. And we know that the head spike fiber binds the capsule that's made out of polysaccharide of the of recipient cells. And near the tail, or at the end of the tail, there are the distal tail hub, megatron, and tail fiber proteins. And these all have putative oligosaccharide binding domains. But interestingly, the megatron protein has a domain that's homologous to a carbohydrate degrading enzyme. And here I'll tell you what we think is the RCGTA binding model. So once the RCGTA particle is close to a recipient cell, usually in an aquatic environment, the first interaction occurs with the polysaccharide that the capsule is made up of. And this interaction occurs mainly with the head spike fiber protein. And this interaction helps the RCGTA particle wheel its way through the capsule towards the outer membrane, where another outer membrane receptor binding occurs with the tail fiber, and that causes a conformational change at the base plate, which causes the insertion of the megatron domain into the outer membrane, causing a pore formation and eventually the release of the DNA through the tail tube. And the DNA is released into the periplasm, and after that, it's factors within the cell that are responsible for homologous recombination of this piece of DNA and expression of it. The main goal of my project is to identify the receptors on Rhodobacter capsulatus cells, the RCGTA receptors. And I hypothesized that this outer membrane receptor is the lipopolysaccharide, or LPS, of these gram-negative Rhodobacter capsulatus recipient cells. And I know there were a few talks about LPSs this morning, but this is a schematic diagram of an LPS molecule. So it has the lipid component called the lipid A, and then there's a short sugar chain that's called the inner core that's usually characterized by the presence of certain sugars. And coming away from the cell, there's another short chain, the outer core, and at the end there's the O antigen, which is characterized by repeat chain units. And not all bacteria has the O antigen. The ones that do are said to have a smooth type LPS, and the ones that don't are said to have a rough type LPS. So with that, I'll get into a brief overview of what my experimental approaches are. So the first set of experiments involve a non-recipient strain of Rhodobacter capsulatus and complementation of this strain with a recipient wild-type genomic library, and then subsequent sequencing of the wild-type fragment. The next set of experiments involve LPS mutants, so identifying potential LPS biosynthesis genes, and then creating using site-directed mutagenesis, creating knockouts of those genes, and then screening those mutant strains for their GTA recipient capability, and looking at their LPS composition. And the last set of experiments involve a transposon mutant library and screening through the library in sort of two steps. The first step is using phage resistance, and the second step is using RCGT-mediated gene recipient capability, and eventually sequencing which gene got disrupted that leads to a reduction in RCGT recipient capability. So getting into the details of the experiments and the results, so I have strain 37B4. It's a naturally occurring isolate of Rhodobacter capsulatus. However, it's incapable of receiving genes via RCGTA particles. So we used a recipient wild-type genomic library to complement this strain. And we were able to endow this strain with recipient capability using this complementation. Which is shown on this bar graph here. So I have the wild type or positive control here. 37B4 has no detectable recipient capability, and the complemented 37B4 does have detectable recipient capability. And when we sequenced the wild type fragment that complemented it, it was shown to contain this previously discovered capsule biosynthesis gene cluster. So we wanted to look at the capsule at the phenotypic level. So we performed Anthony's capsule stain and some phase contrast microscopy. Yeah, these images look terrible on the screen. Sorry about that. The wild type cells, which are known to have the capsule, do have a clear zone around the cells, and this is indicative of the presence of a capsule. Strain 37B4 does not have a capsule because there's no clear zone around the cells. And a complemented 37B4 again has a clear zone similar to the wild type. So this sort of led to the rediscovery of the importance of the capsule in RCGTA binding. But it's also indicative of the fact that there are additional factors that even complemented 37B4 is missing that are involved in RCGTA binding. And that's where other experiments come in. So, so in terms of the LPS mutant strains, the fact of the matter is that the wild type strain that we use in our studies, SB1003, we don't have any data on the composition of its LPS. But what we have from previous papers, like papers from the 70s, from the 80s, is some compositional data of the LPS of some related strains. And two of those strains were shown to have a trisaccharide containing galactose, glucose, and uraminic acid on the core of their LPS. So we figured that if we could go through the genome sequence of our wild type strain and find galactose and uraminic acid biosynthesis genes, those might be potential targets for creating LPS mutants on this strain. And we did find the galactose genes on our genome sequence, which are conveniently placed next to genes that are probably involved in LPS assembly / transport. And we also found uraminic acid biosynthesis genes. So we went ahead and got started with the work for creating a GALU mutant and a new A mutant. And we already have the GALU mutant at hand, and I'm going to show some preliminary data with that mutant. So this graph is showing the relative recipient capabilities of the wild-type strain, SB1003, and the Delta GALU mutant. And as you can see, the recipient capability went down by like 95%, and that would have been great. However, we had a concern. So these are what we call pellet assays. These are quick ways to gauge whether or not a strain has a capsule. So this is the wild type, which I just showed on using the Anthony stain, has a capsule, and this forms a large loose pellet. And delta galU is the opposite. It has a small tight pellet, which is indicative of a loss of capsule. So we asked the question, is this reduction in recipient capability due to the loss of the capsule, or is it because the LPS was truncated? And so we had to bring in a couple more strains into the scene. So the Delta GTAI strain was pre-existing. It's our go-to capsule minus strain. Essentially, it is a quorum sensing mutant, but it ends up losing the capsule. So what we did was create the GALU mutant on the Delta GTAI capsule minus strain. And then we performed GTA assay to evaluate their relative recipient capabilities. And the good news is that even the capsule minus delta galU strain has almost zero recipient capability. It is much lower, or it is significantly lower than the capsule minus GTAI background, as well as lower than just delta galU. So this made us confident that the reduction in recipient capability due to the mutation in the GALU gene is not just because of the loss of a capsule, but because some other receptor is being changed, truncated. But of course, there are no guessing games in science. So we are right now getting prepared to send the LPSs of these strains for mass spec. And in the meantime, we're also going to run some gels and do some silver staining to see if the differences show up on the gel itself. And if not, we'll wait for the LPS mass spec data. So moving on to the third set of experiments, which involve a Rhodobacter capsulatus wild type transposon mutant library. And this mutant library is subjected to phage infections, different kinds of phages. The isolates that the mutant strains that are resistant to each phage are selected for, and those isolates, the mutant phage-resistant isolates, are passed through a second screening of a gene transfer assay to evaluate their relative recipient capability. And isolates that have lower recipient capability than the rest, such as isolate C here, will be used for sequencing purposes to figure out which gene was disrupted that led to the lower recipient capability. So we did all of the wet lab work with a Rhodobacter capsulatus strain called Titan. And these are some of the isolates we screened for their relative recipient capabilities. And we decided that we wanted to sequence these 5 isolates. And we did that, and we found this gene, 3320, from all 5 isolates that we tested. And this is an AlphaFold model of the gene. And when we ran this on HHPRED, this was the top hit, which is the lab A protein from the cyanobacteria Synechococcus elongatus. And this lab A protein regulates the phosphotransferase Chi-C, and these are all part of the circadian rhythm of the cyanobacteria. So it is safe to assume that the 3320 protein performs a similar regulatory function in Rhodobacter capsulatus as well. But because I'm specifically interested at the receptor level, we plan on doing the same LPS analyses on this Rhodobacter capsulatus lab A mutant as well. So I'm almost at the end of the presentation, but to summarize before I go, from the complementation study, we rediscovered that the capsule is important in RCGTA binding, but it was also strongly indicative of the fact that there are other factors that still need elucidation. The next set of experiments led us to the discovery of the GALU gene, which seems to be truncating an additional receptor other than the capsule, and we are eagerly waiting on the LPS results for that strain, as well as waiting on the new A mutant to see if that causes reduction in recipient capability, and to look at the LPS of that strain as well. And lastly, we identified a putative regulatory gene in Rhodobacter capsulatus that has led to lower recipient capability, and that may or may not have an effect on the receptor. And we are also waiting for the LPS analysis for that strain. Overall, there's a lot of LPS analysis for me this year. Yeah, hopefully I get to present those results at some point in the future. That's the end of the presentation. Thank you for listening. Thank you to the organizers of the conference, and special thanks to my supervisor, Dr. Tom Beatty from the Beatty Lab, and my colleagues Yasser and Ali, and our collaborators here at York University, especially Dr. Paul Fogg and Dr. Pavel Barty.